But the problem here is that unlike these limitations that might be associated with adult stem cells, they are hard to differentiate because they are master stem cells. They are primitive in their nature. Especially, in fact, chondrogenesis. It seems that if you are to follow this differentiation pathway and program of gene expression, you will find that you have to start with the zygote with the ES cells, and then the cell will have to decide to become endoderm or mectoderm, and then mesoderm basically what gives rise to the somite, and then the somite will have to become lateral plate, and that has to develop into somatic mesoderm until reaching a stage of a chondrocyte precursor or mesenchymal stem cell-like cell, and that will give you the chondrocytes. So we are basically trying to recapitulate the whole period of growth of that of the person in the test tube, and that's really a bit of a challenge. But scientists basically managed to elucidate a lot of the signaling pathways associated with this decision making that the stem cell has to make, and a lot of that is done through the knockout and knockdown studies on the mice, where, for example, here it's possible to even know which gene is important for the next step in the development of the mouse, and that applies if you want to look even downstream towards formation of a tissue, in this case cartilage. So in our case, the options were either to derive MSC-like stem cells from pluripotent stem cells, or in fact directly drive the chondrogenesis of pluripotent stem cell colonies. So pluripotent stem cells grow as colonies, whether embryonic cells or induced pluripotent cells, and therefore basically bypassing the needs for mesenchymal stem cells. We still think that using the 3D scaffolds would be important in providing maximum signals for driving chondrogenesis and stimulation, leading to cartilage engineering. So again, I'm glad to report that we are making progress with that, where in fact we developed a method, a staged method, where we can drive these cells bit by bit, learning from the signal of development until we reach a population of cells we believe is we've characterized as in fact MSCs, so it shares most, if not all, the markers that we found on the adult MSCs. It has the multi-potential, and most importantly, it has the ability to make cartilage. You can see here when we compare it to our golden standard of adult stem cells, we have still a long way to go, but we are really working on this, and in fact, we have some better results using different biomaterial than this one. But that's not really where we are trying. We are attempting as well to drive the differentiation of the colonies, and you can do this again by modulating, by driving the embryonic cells or pluripotent cells down the mesoendoderm, then the mesoderm, which leads the chondroprogenitors. This is some of the growth factors suggested to be used, and again, my Saudi PhD student Zamani just told me, in fact, this morning that she managed to repeatedly reach this point using, in fact, an induced pluripotent stem cell line that we obtained from Oxford. That cell line has just published a paper in Blood on the generation of B lymphocytes using that cell line. So it is possible, in fact, we are basically heading this way. I don't like this way. I prefer the MSC pathway because with this mechanism, you may still end up with some undifferentiated cells, and these undifferentiated cells may transform, in fact, if you implant them. So you have to purify here, while in fact, if you reach an intermediate stem cell-like, mesenchymal stem cell-like population, then you might be, in fact, having a safer option. Finally, I will take you to the other end. So we talked about here, hopefully alleviating the symptoms and, if you like, keeping somebody from reaching the end point of surgery and arthroplasty and the pain that comes with it, and take you to something that we celebrate always in Bristol after what happened 2 years ago. So building on the technology that we developed and this really intricate signaling sequence we developed for articular cartilage repair. We've been approached by a team in Spain, by Professor Paolo Macriani, who had in fact a patient, and that patient seems to suffer from bronchomalacia, which is narrowing of the bronchus. And what happened here is that the patient was treated in every clinical way possible to save her, but she couldn't. Basically, she would make 2 steps and she would fall down, and she was about to die. That surgeon basically was really brave. He was Italian again. They seem to always be brave to do things like this. And this is where we approached this basically, and they thought, well, can we engineer a trachea using your system, using your controllators? So you can see here, this is an X-ray of her trachea. You can see here the windpipe so narrow compared to the right-hand side. And if I show you, in fact, some bronchoscopy, it will be probably more evident. So this is basically bronchoscopy of the patient. She's called Claudia, and you can see here the left bronchus basically being too narrow compared to the right bronchus. They've done a lot of stenting, and technically the only thing that remained was to remove the entire line. So let me now show you a video showing, in fact, I think there was a lot of details here, but because of time, and I'm happy to discuss the scientific details, and you're most welcome to read the paper if you haven't. But basically, the technique goes like this. So this is a cartoon generated by our collaborator in Spain. So this is Claudia, and you have here her windpipe, which is basically narrowed. And what we managed to do was to reach out to a donor, of course a cadaver here, and she was not compatible. She's not immunologically compatible. And her windpipe was removed. And then that windpipe underwent a process we call is decellularization. And in that process, what the scientist, again, can do is remove all the cellular components, all the antigens of that trachea, and leaving, in fact, the tissue intact. And then we've taken from Claudia, the recipient, stem cells from the bone marrow and epithelial cells from the bronchus, where the stem cells will be seeded on the outside to rejuvenate the cartilage rings. So these are cartilage rings. And the epithelial cells will be seeded inside to form the epithelial lumen. So that was done. Basically, then that piece of cartilage was sent back to Paolo, where he went back to Claudia and removed that narrowing and basically replaced it with the new engineered trachea. So we were very pleased with that, because that proved that, in fact, we moved from the area of stem cells as regenerative medicine into stem cell medicine. So it is, in fact, possible to use stem cells and, in fact, save a life, not just, hopefully, just repair a knee. So this is basically the outcome here. So you can see now, this is the new implant. This is a CT of the implant. And this is now the bronchoscopy here of the tracheal implant here. And you can see it's beginning to revascularize. So this is 4 days after implantation. And the most important thing to me, in fact, believe it or not, was that there was no new rejection. So this process, basically, you can imagine basically what this means. It will transform the field of transplantation, and people now beginning to think of colon decellularization, thinking of, in fact, aorta decellularization, and that will overcome a lot. In fact, some people are going even to xenografts using animal tissue, but that's probably far-fetched, I think, at the moment. Claudia is doing well, and although it's N = 1, in fact, there was another patient in Great Ormond Hospital in London, a young boy, aged 11, who was treated again. Paolo is withholding from treating patients because he's following Claudia very, very carefully to try to improve that technology, but there are many patients lined up for that. So it's really been successful, and it's really been good. So the conclusions, I think that there is progress to real clinical application of cartilage engineering is real and achieved, and that, you know, cartilage implants for osteoarthritis patients are possible. And that hopefully with the scale-up of large cartilage engineering, we can in fact probably resurface a whole joint and save those people with advanced osteoarthritis from needing arthroplasty. And that, in fact, the use of pluripotent stem cells will provide us with an off-the-shelf allogeneic source that we can use indefinitely to treat patients. I wish to thank my team here, and there is in fact a student here standing missing. We don't have his picture here. This is Amani, who is working with the iPS cells. This is Nandita, my postdoc, and this is my technician and PhD student. And this is Anthony Hollander, Sally, Trevor Sims, and Sanjay Mistry. They are all co-workers working closely, and of course our funding bodies who help support this work. And thank you for being a good audience. Thank you so much.
Thank you, Ed.